In this session, we will discuss microbial metabolic products and their enhancement of production. So, this is the learning objective. After the session, you will be able to understand, describe and write about different types of metabolic end products and how can we increase the different types of metabolic end products in an industrial scale. So, what is meant by metabolic end products or microbial metabolic products? So, microorganism as such is an miniature of biochemical factory which is capable of converting simple substrate into vast variety of metabolic product through enzymatically controlled biochemical reactions. Thereby, it will play a vital role in the industrial biotechnology, pharmaceutical industry and food and agricultural sector. So, as we all know, the fermentation is a process of growing a microorganism in a substrate in an optimum condition by giving the essential nutrients and by giving optimal conditions of pH, temperature, aeration, etc. So, we know that in a closed system, microorganism will show a specific pattern of microbial growth that is known as growth curve. There is a lag phase, then there is a log phase or exponential phase, then an stationary phase and then a decline phase. So, during these 3 or 4 phases, especially in the 2 phases, there are different types of metabolic end products which will be produced during their growth. So, basically the metabolic end products is classified into primary metabolites and secondary metabolites. So, as the name indicates, primary metabolites are the metabolites which are essential for the growth and energy of the microorganism itself, and secondary metabolites, they are non-essential for the growth, but it will confer the ecological and competitive advantage, like it will produce antibiotics. So, this is the classification. We have primary metabolites and secondary metabolites and some bioconversions. And in primary metabolites is again divided into essential metabolites and metabolic end products, which means these are the primary metabolites, but some of the metabolites are essential for the growth of microorganism like amino acid, nucleoside and vitamins, and some primary metabolic end products which even though they are not essential, they will be produced like ethanol, acetone, lactic acid and butanol. And there are some secondary metabolites, these are not essential for the growth of microorganism, they are the end products. So, antibiotic, alkaloid, gibberellin, pigment, and we will use the same microorganism for the bioconversion. We have already studied what is meant by microbial biotransformation reaction. There are different reactions like hydroxylation, oxidation, hydrogenation or dehydrogenation. There are different microorganisms which have an ability to carry out this kind of reactions. So, we will use this kind of microorganism for the production of steroids, amino acids as well as ascorbic acid. We have already studied in the unit 4 what is meant by microbial biotransformation reaction with the special emphasis on the production of steroids from stigmasterol. So, we have already studied that part. So, here we will discuss what is meant by primary metabolites and what is meant by secondary metabolites. How can we increase the yield of the primary metabolites and secondary metabolites. So, first one is primary metabolites, also known as trophophase, because usually this will be produced in a balanced growth of microorganism. It means as the required nutrition is there for the growth of microorganism, and there is no metabolic waste product which is produced in the fermenter, this kind of metabolites will be produced and occur when all the nutrients are needed by the microorganism are provided. Only if we provide all the essential requirements are provided. So, this is the best way to increases the yield of primary metabolite. We have to give the essential requirement whenever there is a deficiency of nutrients in an fermenter. Okay. Then these are essential for the vital existence of the microorganism as well as reproduction of the cell, and it is again divided to primary essential metabolites like vitamin, amino acid and nucleosides, and primary metabolic end products, normal and traditional end products of the fermenters like ethanol, acetone and lactic acid. Now, how can we increase the overproduction? How can we overproduce these primary metabolites? So, basically by eliminating the feedback inhibition. So, by using some kind of auxotrophic mutant, which with block in one of the steps in the biosynthetic pathway, which is concerned with the fermentation of primary metabolites. So, by using some kind of mutant microorganism, we can increase the primary metabolites, or by increasing the essential nutrients. Mutant microorganism with an anti-metabolite resistant, which will exhibit defective metabolic regulation, also can be used to increase the overproduction of primary metabolites. Okay, so what is mean by primary metabolites? These are the metabolites which are produced during the log phase of the growth curve. During the log phase. So, what is the peculiarity of the log phase or specificity of log phase? Which means there will be the sufficient amount of nutrients for the growth of microorganism. There will be sufficient amount of nutrients for the growth of microorganism. As the growth of microorganism increases, the primary metabolites also will increases. So, either by increasing the concentration of the nutrients, we can increase the primary metabolites, or by using some kind of auxotrophic mutants or mutant microorganism with the anti-metabolite resistance. Okay. Now, coming to the secondary metabolites, as the log phase end and it will enter into the stationary phase, it is known as, that kind of phase is known as idiophase. So, these are the secondary metabolites, are the metabolites which are produced during this idiophase. So, that is why it is also known as idiopathic metabolites. And these are not essential for the growth of microorganism. Examples like antibiotics, steroids, alkaloids. But there are some specificity, only the few microorganism will produce the secondary metabolites. Not every microorganism will produce, only the few type of specific microorganism will produce secondary metabolites, and some of the environmental factor also will influence the production of the secondary metabolites like temperature, aeration. All these factors will affect the production of secondary metabolites. So, sometime what will happen during the fermentation process, in the production process we will mention we have to keep the temperature of some specific temperature for these days, and after that temperature have to be increased for several days. So, there will be variation in the temperature during the entire process of fermentation. This is for this kind of purposes, for increasing the secondary metabolites. First, we have to increase the growth of microorganism. Then we will increase the secondary metabolite by changing some environmental factor. And some of the microorganisms will produce some group of secondary metabolites, not only one metabolite. Sometimes they will produce a group of secondary metabolites, and sometimes the biosynthetic pathway are not clear. Unknown ആയിട്ടാണ് അതിന്റെ പ്രൊഡക്ഷൻ ഉണ്ടാവുന്നത്. എക്സാക്ട് നമ്മളൊരു അതിന്റെ കെമിക്കൽ സ്ട്രക്ചർ എങ്ങനെയാണ് മാറുന്നത് എന്നൊക്കെ ചോദിച്ചു കഴിഞ്ഞാൽ പലപ്പോഴും fermentation is a complex process, so we cannot predict the biosynthetic pathway, which compound is converted to next. We cannot predict that. Finally, we will get some essential compound. We have to purify that. Now, how can we increase the secondary metabolites? By induction. So, by sometime in our production process, other than the substrate used for the production, we will mention some kind of inducers has to be added into the fermenter. So, first one is methionine. Methionine is usually added to increases the production of cephalosporin, and second one is tryptophan, which is usually added to increases the ergot alkaloid biosynthesis. So, these are some of the examples of induction. And second one is end product regulation. Most of the case, especially in case of antibiotic, the end product which are produced during the fermentation process as in secondary metabolites will inhibit the further production of the secondary metabolites, because most of the time the secondary metabolites are antibiotic, especially penicillin, streptomycin, puromycin, chloramphenicol. All these are some kind of broad spectrum antibiotic. So, it will inhibit almost all kind of microorganism. So, it will inhibit the selected microorganism which is used for the fermentation. Okay, so that's the end product regulation. So by either we have two options to increases the this kind of end products. Either we have to remove the secondary metabolites which is formed during the fermentation, so that it will not inhibit the microorganism, or we have to use some kind of microorganism which have a resistance towards this antibiotic or substrate which are produced during fermentation. So that are the two methods to increases the secondary metabolites end product regulation. Now next one is catabolite regulation, which means in every biosynthetic pathway there will be a key enzyme which is involved for the production of secondary metabolites. So by inhibiting that key enzyme, we are not inhibiting any other kind of microorganism or anything, we are just inhibiting that key enzyme which is there for the biosynthetic pathway of the specific secondary metabolites. So either by inactivating or by inhibiting or repressing by adding some commonly used substrates like carbon or nitrogen sources. We are not going to add anything other than this. We will add the large amount of glucose for inhibiting several antibiotics. Glucose as such we are using as a carbon source for giving the carbon skeleton as well as for the energy. But if we are increasing the amount of the glucose in a fermenter substrate or media, it will inhibit the production of several metabolites. So by specifically adjusting the enzyme which is used for this kind of catabolic pathway, we can increases the secondary metabolites. And last one is phosphate regulation. Inorganic phosphate itself is an very essential for the growth multiplication of the many microorganisms. By increasing the concentration of phosphate, we can increases the production of the secondary metabolites. Okay, so these are the different methods for the production of secondary metabolites. So this is the basic difference between primary metabolites and secondary metabolites. Usually the primary metabolites are produced during the log phase and secondary metabolites are produced during the stationary phase. So in order to increase the primary and secondary metabolites, we have to prolong that phases. For increasing the primary metabolite, we have to prolong the log phase. To increases the production of the secondary metabolite, we have to prolong the stationary phase. And primary metabolites are essential for the growth, and secondary metabolites are not essential for the growth. And production pattern are usually associated with the growth of the microorganism in case of primary metabolites, which is not growth associated in case of secondary metabolites. And common to many microorganisms, the metabolites will be produced. Secondary metabolites, as I mentioned, only specific kind of microorganism will produce secondary metabolites. An example, ethanol, amino acid, organic acid. Secondary metabolites examples are antibiotic, alkaloids and pigments. And basically primary metabolites are mainly used for the food and supplement purposes, and secondary metabolites basically for pharmaceutical as well as pesticides. Most of them are have an antimicrobial activity or antibiotic like substances. Okay, so that's all about the primary and secondary metabolites and their enhancement of production.